Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 4 for January 2023. Let us begin with the first question. Question 1. The resistance of bacteria to antibiotics can be studied using a mass string. A mass string is made of filter paper. Each part of the mass string contains a different type of antibiotic or different mass of the same antibiotic. The diagram shows a mass string placed on agar in a petri dish that has been spread with bacteria. The table shows the mass of each antibiotic in the mass string. So we can see this is the mass string. We see the different antibiotics that have been used. This indicates the clear zone, and this is the region where bacteria have successfully grown. This table shows part of a mass string and the different antibiotics that have been used, as well as their concentrations or compositions. Beginning with antibiotic A, we can see this is not effective at all because there is no clear zone, as well as on this one and this one here. But we can see there is a clear zone here, there, and there, so there is some degree of efficiency. However, among the 5 effective ones, this is the least effective. And then we can see these have the same effectiveness. The corresponding concentrations here have the same effectiveness, meaning we can take the one with the lower concentration, since increasing the concentration did not change the effectiveness of the antibiotics. I'm talking about comparing B2 and B3. We can see we have a higher concentration here, which is 0.5 milligrams, instead of 10 micrograms. This one here has a lower effectiveness in comparison, and E also has a lower effectiveness in comparison to B2 and B3. So down here they say, clear zones around the mass string indicate that the growth of the bacteria has been inhibited. The greater the diameter of the zone of inhibition, the more effective the antibiotic. So here they say, state 2 precautions that should be taken to ensure that only one type of bacteria is grown in this culture. To ensure there is no contamination, we have to use aseptic techniques. So you have to work besides a Bunsen flame. You have to use sterilized equipment and also sterilize the workplace. And you have to cover the petri dish to minimize the time of exposure to the air that could contain contaminating microorganisms. In part B, how much more antibiotic is in part B3 than in part B2? So for this, we have to go back comparing B3 and B2. If you look at B2, there is 10 microgram, while in B3 we have 0.5 milligrams. You know 1 microgram is 10 power negative 3 milligrams. So the 10 micrograms should be 10 power negative 2 milligrams, which is corresponding to that. While in B3 we have 0.5 milligrams. So we're going to get the ratio of these two. So the ratio of B2 to B3 should be 0.01 to 0.5, which is 1 to 50. And therefore, it's going to be 50 times more. So C is the answer. In part C, explain how the results shown in the diagram can help a doctor decide how to prescribe antibiotics to a patient who is infected with this type of bacterium. So here we have to go back to the mass string. We can see this has no effectiveness at all. These have low effectiveness in comparison to these two. However, because this and that have the same effectiveness, here this has a higher concentration, it's going to be preferred to prescribe the one with a lower concentration but high effectiveness. So we will prescribe B2, and we will not prescribe A, C1 as well as D. So here I said, antibiotics A and D should not be prescribed because even at a mass of 100 micrograms, there was no clear zone. Antibiotic B is the most effective because all masses used had a degree of antibacterial effect. So the doctor should prescribe antibiotic B with a mass of 10 microgram since higher masses did not show improved antibacterial effect. So the one with 10 microgram, of course, is going to be antibiotic B2, which we saw here, this one here. So this brings us to the end of question 1. Let's continue to question 2. Question 2. Some bacteria are photosynthetic. The diagram shows a photosynthetic bacterium, which we can see here. So they say which labeled structure contains the gene coding for the enzymes involved in photosynthesis. This is going to be where the DNA is found, and that is going to be in part R. So R was my answer. You can see P is going to be the ribosome, then Q is the cell wall, and then T is the membrane. And down here they say, where do the light-dependent reactions take place? They take place in part T. Remember, this is a bacterium, so they take place in part T, which is going to be the membrane. And then they ask, where do the light-independent reactions take place? the light independent reactions will take place in the cytoplasm, which is going to be region S. So here, B is my answer. Moving on. In part B, the graph shows the absorption spectrum of photosynthetic pigments in a plant and in a bacterium. So we have the plant, you can see that, and the one for the bacterium down here. When we compare the results, we can see there is absorbance over a smaller range here in comparison to that in bacterium, where we have over this region and again this region. So bacterium have absorbance over a wider range, and we also see the absorbance is higher. While this is below 0.7, this is already above 0.8. So higher absorbance here and absorbance over a wider region in comparison to the plant ones. So moving on. Describe two conclusions that can be made about the differences in absorption spectra shown in these two graphs. Of course, this is similar to what I've said. Bacterial pigments can absorb light over a wider range of wavelengths than plants, and then bacterial pigments can absorb more light at more wavelengths than plants. This is what I've shown here. Over more wavelengths, we can see absorbance of a wider region or wider wavelengths, and here we see absorbance of a smaller region of wavelengths, and then we see this is higher in comparison to that. So lastly, I said for plants that the greatest absorbed light is at 440 nanometers, and for bacteria it is at 380 and at 880 nanometers, which we can see here. There is this one here, which is the highest there, while for bacterium we have that and that, which are about 380 and about 880 nanometers. Moving on. In part C, photosynthetic pigments can be identified using chromatography. The diagram shows part of a chromatogram. So this is the chromatogram. We see the origin. There is a pigment, and they've told us the direction of movement of the solvent front. Then they say, measurements are taken from a chromatogram to determine the RF value for the photosynthetic pigment. Describe the measurements taken and how they can be used to calculate the RF value for this pigment. Remember, RF is distance moved by the sample divided by the distance moved by the solvent. So we have to measure the distance moved by the pigment from the origin, and we also have to measure the distance moved by the solvent from the origin, and then we divide them in this manner in order to calculate RF. So measure the distance moved by the pigment from the origin, and measure the distance moved by the solvent from the origin, and calculate RF as below here. So this brings us to the end of question 2. Let's continue to question 3. Question 3. The photograph shows part of a vanilla plant grown in tropical areas along the Indian Ocean. So we see the flower as well as the developing bean pod. They say vanilla is an expensive spice to produce due to labor costs and damage to crops by pests. Viruses such as vanilla mosaic virus damage the leaves of vanilla plants. The vanilla mosaic virus has a similar structure to tobacco mosaic virus, which is TMV, which describes TMV. TMV is an RNA virus and it has no envelope, so here D is going to be our answer. Moving on. In part B, disposing of plastic waste is a global problem as it is very difficult to recycle. In 2018, 8.3 times 10 power 12 kilograms of plastic was produced. Only 8.7% of this plastic was recycled. Calculate the mass of plastic that was not recycled, and they want you to express your answer in standard form to one decimal place. So if 8.7 was recycled, that means the percentage of not recycled is 100 minus 8.7, which we did here. So not recycled is going to be 100 minus 8.7 divided by 100 times the total, which gave us that. And my final answer was 7.6 times 10 power 12. This is to one decimal place and in standard form. The next part says, scientists are developing methods that use microorganisms to break down plastic. Some of the products produced are being used for other purposes, such as vanilla flavoring. Explain why some microorganisms can break down plastic. Breaking down plastic requires enzymes, so these microorganisms release enzymes that hydrolyze the bonds in the plastic in order to break it down. Moving on. Here they say, one type of bacteria, Pseudomonas, uses half of the plastic it breaks down to produce its own biomass, the rest released as carbon dioxide. Explain how the breakdown products become biomass and carbon dioxide. So here the hydrolyzed products are going to be absorbed into the bacteria, and then respiration is going to be carried out where glucose is going to be used to produce ATP, and this process leads to CO2 production. So the other organic compounds like amino acids, lipids, and excess glucose are used to make molecules like glycogen as well as proteins that are used to make the biomass form inside the bacterium. So this brings us to the end of question 3. Let's continue to question 4. Question 4. Natural rubber is harvested from rubber trees. The photograph shows rubber being collected from the trunk of a rubber tree in Thailand. So this is the process of collecting the rubber. In part A, the graph shows the mass of rubber produced in Thailand in 2019 and 2020. They say in 2020, Thailand produced 4.37 million tons of rubber. Estimate the mass of rubber produced in Thailand in 2019. I used the ruler to get the measurements. So this height is about 6.4 centimeters and this is about 5.8 centimeters. If this was produced in 2020, it means 5.8 centimeters corresponds to this amount. So if 5.8 centimeters corresponds to that, it means 1 centimeter corresponds to this amount. So the 6.4 centimeters are going to correspond to this amount times 6.4, which gave me 4.822 times 10 power 6. Therefore, my answer should be that in tons. In part B, rubber is synthesized by specialized cells in the trunks of rubber trees. The diagram shows the structure of one molecule that these cells use to synthesize rubber, and this is the molecule. They say galp is produced in the light-independent reactions in the leaves. Explain how the cells in the trunk are able to synthesize the molecules in the diagram using galp produced in the leaves, and they want you to use the information in the diagram and your own knowledge to support your answer. If we look at this molecule, there is carbon and hydrogen as well as oxygen. These could come from photosynthesis. However, there are atoms of phosphorus, and these are going to come from phosphates. So here I say galp is converted into glucose, and the glucose is converted into sucrose, and this sucrose is going to be transported through the phloem to the storage sites. Also, phosphates are going to be delivered from the soil by the xylem tube through active transport. Then the molecules in rubber are synthesized by incorporating the phosphate ion. All these processes will use energy that is provided by ATP from respiration. All these processes require enzymes to participate in the formation of bonds within the molecules of rubber. Moving on. In part C, a rubber tree can be used to harvest rubber until it is about 32 years old. Explain how dendrochronology could be used to determine the age of a tree that is still producing rubber. Because this tree is still producing rubber, it is not going to be cut down, so we'll need to remove a sample from the tree trunk. This could be done by boring into the tree trunk, and then we use the sample to count the number of rings. The number of rings counted should equal to the age of the tree, since new rings are made each year. So this brings us to the end of question 4. Let's continue to question 5. Question 5. Global warming is one consequence of anthropogenic climate change. The diagram shows a model of some of the effects an increase in temperature of either 1.5 or 2 degrees Celsius could have on the environment. So here we have increasing impact from 1.5 to 2 degrees Celsius. It could lead to loss of habitats. This could be in plants as well as vertebrates. Population exposed to water scarcity. We can see extreme hot days, population exposed to drought, and sea level rise. So the first question says, explain why this diagram is described as a model of the effects of an increase in temperature. This is a model because it predicts or shows what will happen in the future based on the information or data collected from the events that have occurred previously. The next part says, calculate the percentage increase in the population exposed to drought if the temperature increased by 2 degrees and not 1.5 degrees. Now, this was quite a tricky question because there is not extra information given to us. And again, remember, they've told us percentage increase in the population exposed to drought. So this is the population we're looking for. If there was a change, instead of 1.5, if we go to 2, I used my ruler and got the ratio of that to that, which is about 1.1 to 1.4. So the ratio of 1.5 degrees to 2 degrees is 1.1 to 1.4. It means if I'm taking the ratios, the increase is going to be that minus that, which gave me 0.3. So percentage increase should be 0.3 divide by 1.1 times 100, which gave me 27.3%. Moving on. In part B, it has been estimated that because humans have planted more trees and crops, the leaf area increased by 0.5 million kilometers squared. China accounts for 25% of this increase. It planted forests and crops in equal proportions. India accounts for 7% of this increase. It planted mostly crops. Discuss the possible impacts of planting forests or crops on global warming, biodiversity, and the local population. So on global warming, planting both trees and food crops provide carbon sinks for carbon dioxide by removing it from the atmosphere. This is due to photosynthesis. This decreases the greenhouse effect and leads to reduced global temperatures. However, planting crops reduces CO2 to a lower extent than forests because harvesting occurs annually. Here we're talking about harvesting of crops that are going to be consumed. It occurs annually and machinery is used. Since this consumes fossil fuels, it will not have a greater effect on removing CO2 from the environment because machinery use is also going to release CO2. When we go to biodiversity, planting crops and forests provides food and habitats for organisms, mainly herbivores. This leads to an increase in biodiversity. However, it could also lead to monoculture. Monoculture is where farmers plant only one type of crop because it's maybe beneficial or it grows faster or it has a higher yield or it gives them a lot of money. So if this monoculture occurs, it will lead to low biodiversity, and also use of pesticides could be harmful to the organisms or to some organisms. When we go to human population, plants will provide food and employment on farms, and this can lead to increase in population. However, crops may be grown on land that could be used for other purposes, like housing. Crop use could be expensive due to the use of fertilizers as well as pesticides, so this could be a negative effect, although we have a positive effect due to employment and so on. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Changes in temperatures affect the rate of enzyme activity and survival of living organisms. Which describes an enzyme in a living organism? So these two are wrong because enzymes are organic, they're not inorganic. D says organic substance that increases activation energy. This is wrong. Enzymes decrease activation energy, so C is going to be our answer. In part B, the graph shows the survival time of one species of fish in water at different temperatures. On the vertical axis, we have survival time, and on the horizontal axis, we have water temperature. So we can see the survival time increases and then remains constant before it begins to decrease. So they say, name the type of scale that has been used on the y-axis. This is a logarithmic scale. The next part says the zone of tolerance is the range of temperature that a fish could survive at for longer than 5,000 minutes. Which two biotic factors could cause the fish to die in this range of temperature? This is a biotic that is abiotic, and this one here is abiotic. So the only two biotic factors are predators and pathogens. So C is going to be our answer. The next part says the body temperature of a fish changes with the temperature of its surroundings. Explain why the survival time of fish is reduced at temperatures lower and higher than those in the zone of tolerance. This is mainly due to enzyme activity. The zone of tolerance, we saw the survival is constant, but below the temperature 0, we can see the survival is going to decrease, and beyond 20 degrees, we see the survival is also going to decrease. The decrease or low survival in this region is going to be due to low kinetic energy, but the decrease or low survival here is going to be due to high temperature causing the enzymes to denature. So here I said, fish survival is maintained by metabolic reactions. These are enzyme controlled. At lower temperature, there is low kinetic energy for the successful collisions to form the enzyme substrate complex. And at a higher temperature, the active sites of the enzymes are denatured and the rate of reaction decreases, so the fish begin to die. Moving on. In part C, enzymes were stored for different lengths of time. The effect on the initial rate of reaction was investigated. The graphs show the results of this investigation. So we have on the vertical axis initial rate of reaction, and on the horizontal axis we have temperature. we see here there is an increase and then a decrease for all graphs, whether enzyme stored for 3 hours, enzyme stored for 10 hours, or 26 hours. But we can see that the higher the storage time, the lower the initial rate of reaction, since 3 hours had the highest, 10 hours the next, then 26 hours had the lowest. So here they say, calculate the Q10 for the enzyme stored for 3 hours. They want you to use this formula, where RT is the initial rate of reaction at 20 degrees. If RT is rate at 20, it means RT plus 10 is going to be rate at 30. So for enzyme stored for 3 hours, I had to find the rate at 30, which is that, and I had to find the rate at 20, which is that. So that is 4.2, and this is 2.7. So Q10 was 4.2 divided by 2.7, which is 1.56, and that is my answer. Moving on. Explain why the optimum temperature for this enzyme is affected by storage time. Now, when they say explain, we have to state the fact and then give a reason why. My fact is, the longer the storage time, the lower the optimum temperature for the enzyme. This is my statement. Then I have to give a reason. And the reason is, as storage time increases, the active sites are going to change shape. So the longer the enzyme is stored, the more the shape is distorted, and the harder it is for the enzyme to regain its shape. I put some extra explanation here. I say during storage, the kinetic energy promotes vibration, so breaking the links between the R groups. And when you store the enzyme for longer periods of time, there is going to be more links that are broken, and this could result in complete distortion of the enzyme, and therefore denaturation can completely occur. So this brings us to the end of question 6. Let's continue to question 7. Question 7. Analysis of DNA samples taken from endangered species can be used in the conservation and management of these species. The diagram shows part of a molecule of DNA, which we have here. So they say, which row of the table identifies the bonds Q and R? Bond Q is a hydrogen bond, and bond R is a covalent bond. So here B is going to be our answer. When we go down here, which is a negatively charged part of a DNA molecule, that is going to be the phosphate, which is S. Moving on. In part B, the Malaya is an endangered bird endemic to Indonesia. The photograph shows a Malaya digging a hole in sand to lay her eggs. So this is the bird. Malayas live in the forest and only come to the beaches to lay their eggs. Once the eggs are laid and buried in the sand, the birds return to the forest. When the egg hatches, the young bird digs its way out of the sand and can fly straight away. Habitat fragmentation is thought to be partly responsible for this species being endangered. The forests are separated from the beaches by activities of humans. State the meaning of the term endemic as used in the context of this question. This means they want us to answer in referring to that bird. So I said the Malaya birds are only found in Indonesia. The next part says, give one reason why habitat fragmentation could endanger these birds. There is going to be human activity between the forest and the beaches. So once the eggs hatch, the birds cannot fly from the beaches to the forest. So I say the newly hatched birds cannot fly to the forest from the beach area. And then PS, remember eggs are laid at the beach and buried in the sand. So once eggs hatch, the birds get out of the sand, but they cannot fly back to the forest. Here they say, suggest the advantage of laying the eggs in the sand. This is going to provide them the optimum temperature. It could be warmth in order for them to hatch in due time. The adults leaving the site. This means if there are no adults, there will be no sight of the existence of the eggs, and therefore humans or any other organisms cannot dig up or look for the eggs in the sand. And then a young bird being able to fly, it enables it to be able to escape from predators in order to fly away to the forest once it has been hatched. Yeah, I said laying eggs in the sand provides the right temperature to make them warm. And then adults leaving the site does not leave, the indication of the eggs being buried nearby. The young being able to fly enables them to escape from predators. Down here they say, in a genetic study of these birds, DNA from the developing chick was isolated from the remains of the eggshell and amplified using PCR. To amplify a DNA molecule using PCR, 2 different primers are used. Explain why two different primers are used to amplify one molecule of DNA in a PCR. This is because the DNA has two strands, so each strand has to be copied, and therefore we need to have two primers. So I say a primer is used so that the DNA polymerase can bind onto a DNA strand. Since DNA has two strands, each strand requires a different primer. Moving on. Here they say, these birds are thought to be monogamous, which means that they only mate with one bird of the opposite sex. Birds are only monogamous if the ratio of male to female is 1:1. In one genetic study, DNA from 23 eggshells was analyzed. The study was that 8 of the chicks were female and 15 chicks were male. Comment on the claim that Malays are monogamous. Remember, to be monogamous, the ratio has to be 1:1. But here, if we can see the ratio of male to females, females are going to be 8, as we can see here, and then 15 are going to be males. So the ratio is 8 to 15, which is 1 to 1.9, or you could say 1 to 2. This ratio is not 1:1, so they will not be monogamous. So the idea that they are monogamous could be because the sample size was too small. So I say it based on the results, there are more male chicks than females. Or you could say the observed ratio of the female to male is about 1:2, and this is different from the expected for monogamy, which is 1:1. So the observed results are different from the expected ratio because the sample size was too small. This part in red is not required to be included. I just put it there for extra explanation. So this brings us to the end of question 7. Let's continue to question 8. Question 8. Dengue fever is caused by infection with a dengue virus. This virus is transmitted by mosquitoes. The virus replicates inside the mosquito and is then passed onto a person in the saliva of the mosquito. The virus then replicates inside the infected person and causes the symptoms of the infection. Symptoms include fever, headache, and a rash. Complications include bleeding, low levels of platelets, and low blood pressure. It is estimated that globally there are 390 million new cases of dengue fever per year. The infection rate is rising. This disease can be fatal. Groups of scientists are working to develop vaccines, treatments, and methods to reduce the spread of this virus. In part A, the dengue virus is an RNA virus. The RNA is translated on the endoplasmic reticulum of the host cell into one polyprotein. Describe how viral RNA is translated. So of course, because we have the endoplasmic reticulum, around here we will have the ribosomes. So this RNA is going to be attached onto the ribosome, and then translation will occur. So I say viral RNA will attach onto the ribosomes of the host cell, and then the host cell transfer RNA containing the anticodon that are complementary to the codon on the viral RNA will carry amino acids to form the polypeptide bonds between adjacent amino acids during the formation of the polypeptide. Moving on. Here they say, the diagram shows these polyprotein and the function of some of the individual proteins it contains. The arrows indicate where the individual proteins need to be separated by protease enzyme, which is that. So we can see there is need for separation to occur at all these sites. We can see until there. We have the protein forming the capsid, protein embedded in the envelope, protease enzyme, and so on. So they say, one group of scientists has found that an inhibitor to this protease enzyme reduces the number of viruses produced in infected cells. Suggest how this inhibitor could result in few viruses being produced. So this inhibitor could bind onto the enzyme, which is going to be the protease enzyme, and this is going to prevent the enzyme from binding onto the polyprotein, which is its substrate. So the peptide bonds in the polyprotein will not be broken, meaning fewer individual proteins will be produced. For example, you can say fewer protein forming the capsid or fewer proteins embedded in the envelope will be formed. So the individual components like the envelope proteins can be assembled into individual viruses. So if one of these proteins is missing, it means the virus particles cannot be assembled properly, and therefore fewer viruses are going to be produced. Moving on. In part B, another group of scientists showed that an extract from the leaves of the papaya plant helped in recovery from dengue virus infection. They showed that the platelet and white blood cell counts were higher in people treated with the extract than in people given a placebo. Explain how these changes may have helped in recovery from dengue virus infection. Because they say the patients had higher platelet and white blood cell counts, the platelets are going to allow for clotting of blood, and white blood cells are going to enable B cells as well as T cells to carry out immune response. So I say higher platelet counts result in more blood clotting, and this leads to less bleeding. Higher white blood cell count leads to increased immune response. These are 4 marks, so we need to give some examples. So I say, for example, B cells will produce antibodies, and the T killer cells can destroy infected body cells in order for the symptoms from the infection to decrease. Moving on. In part C, another group of scientists infected the eggs of mosquitoes with Wolbachia bacteria. The Wolbachia do not harm the mosquitoes but compete with the virus so that it is harder for it to replicate. The bacteria can also be passed on to future generations of mosquitoes. One study in Indonesia showed a 77% reduction in new cases of dengue infection after the introduction of Wolbachia into the mosquito population. They want you to describe 2 advantages of this method of disease control. This is a biological control, and the advantage is fewer people are going to die from dengue because using a bacteria is an effective method, and also because the bacteria is passed on to future generations, so we can see future generations of mosquitoes who possess the bacteria, thereby saving lives, and actually will not repeat the process of reintroducing the bacteria. Then this is a biological method, so mosquitoes are not completely destroyed. Remember, mosquitoes also have their own advantages in the food chain, so mosquitoes are not completely destroyed, so no interruption on the food chain. Question 9. Infections with Mycobacteria causes TB, which is one of the top 10 causes of death in the world. In 2019, an estimated 1.2 million children, 3.2 million women, and 5.6 million men developed TB. Calculate the ratio of children to women to men who develop TB. So the ratio is going to be 1.2 times 10 power 6, or you could say 1.2 million to 3.2 million to 5.6 million, which is 1 to 2.7 to 4.7, or you could use ratios in this manner as well. So I chose this, and that is my answer. In part B, explain how infection with Mycobacteria tuberculosis may result in death. Of course, we know during tuberculosis some tubercles are going to form, and this is going to lead to the destruction of lung tissue, thereby decreasing the surface area for gaseous exchange. So less oxygen will be available for gaseous exchange. They could also be opportunistic infections due to the weakened immune system, and this could lead to death. Moving on. In part C, Mycobacterium tuberculosis has evolved to evade host immunity by interfering with macrophage function. In one investigation, scientists showed that Mycobacterium tuberculosis can inhibit the activation of a process in macrophages. This inhibition results in less of chemical IL-1 beta being released. The graph shows some of the results of this investigation. We can see uninfected untreated macrophages. We see this is less. Untreated macrophages infected with Mycobacterium tuberculosis. Uninfected macrophages treated to stimulate IL-1 beta release. We can see the concentration is high. And macrophages treated to stimulate IL-1 beta release and infected with Mycobacterium tuberculosis. Explain why the macrophages have to be treated to stimulate IL-1 beta release in this investigation. They want you to use the information in the graph to support your answer. Now, stimulation was because the untreated macrophages did not release IL-1 beta, so inhibition of IL-1 beta production by the macrophages due to infection could not be shown. To study the inhibition, and here they say, an ELISA is used to detect a specific antigen. The IL-1 beta was detected using an ELISA. So step 1, they say antibodies attached to plastic container, which is that, and in step 2, the container is rinsed with water. This rinsing process removes any unattached antibodies. Then in step 3, IL-1 beta added to the container. This is going to be attaching onto the antibodies. And then step 4, the container is rinsed with water. This ensures that the unattached IL-1 beta are going to be washed away as well. And step 5, antibodies with a dye attached to them added to the container. This could be for visualization. So here they say, explain how the steps in the ELISA result in the identification of IL-1 beta. They want you to use information in the diagram to support your answer. So I said antibody 1 is used to attach to the plastic so that everything does not wash away. And then antibody 1 and 2 are both complementary with IL-1 beta because each attaches on one side of the IL-1 beta, so they can bind to different sides of IL-1 beta. Also, the dye attaches to the antibodies so that they are visible during visualization. The washing process gets rid of unattached antibodies so that only attached antibodies and antigen remain. The key thing here is antibodies as well as the antigen. In this case, this is acting as the antigen, so anything that is not attached is going to be washed away, and that is the reason as to why we use washing with water. Moving on. The scientists identified a protein that can be responsible for inhibiting the activation of the macrophages. They used Mycobacterium tuberculosis with a mutation in the gene coding for this protein as part of the investigation. This involved comparing the growth rates of these mutant bacteria with the unmutated bacteria. They want you to describe how optical methods like turbidity can be used to compare the growth rates of these two populations of bacteria. So here, because it's culturing, of course, we have to use aseptic techniques to culture both types of bacteria using similar conditions. These similar conditions could be the medium used, the temperature used, the pH, and so on. Then, because we are looking for optical methods, we have to use a colorimeter. So you could use a colorimeter and measure the absorbance, or you could measure transmission or transmittance of light through the sample. You need to ensure that a calibration curve is drawn using absorbance or transmission results accompanied with a hemocytometer. This enables us to draw the calibration curve. Then you will plot a graph of number of bacteria against time and calculate the growth rate by finding the gradient of the tangent at a point on the curve. Then you can use a suitable statistical test like the t-test to compare the results. And again, remember, we are comparing the growth rate, so there could be a suitable statistical test that you can use to compare the results. Then next they say, suggest how this protein released by Mycobacterium tuberculosis could be used to develop a treatment for TB. The released protein could be used as a vaccine. The vaccine could be injected into the patients, and then the patients can produce antibodies as well as memory cells. So this brings us to the end of question 9 as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.